Hi everyone. In the earlier module, we learned about the data management plan, which is a key plan involved in the data management process. So in this module, we are going to look about three other guidelines, includes data validation guideline, non-CRF data guideline, and data transfer guidelines. So data validation is the process of testing the accuracy and the quality of data obtained throughout the study, which should be in compliance with the study protocol requirements. Data validation guideline is a document which deals with the activities that brief about the procedure of study-specific validation and query management throughout the study. The requirement of the guidelines are as follows. Screen testing. So we already discussed about the screen testing. Once the annotated document is prepared and ECRF is also uploaded in the test environment in draft version, and the same is tested against the ECRF requirements as well as with the protocol. So this screen testing is also an important part. Then next comes the data check specification document, which is going to be a time saver while performing the data entry, because all logical type of discrepancies would be identified and uploaded to the system, and same needs to be tested. And validation procedure, which tells about how these things are to be validated. Global ruling we'll discuss in the further slides. Flow of discrepancy note, what would be the flow and how the discrepancy notes needs to be updated. Review of critical data, how to handle the unresolved discrepancies, finalization and revision of this guideline. If you see, the data validation guidelines also mentioned in a chart flow diagram here. Screen testing is a part, error check validation document, how the procedure is going to be enabled for the validation process, global ruling, flow of discrepancy, review of critical data, unresolved discrepancies, and finalization and revision. So first part is screen testing. So how it is done? Once an ECRF is designed, it is to be validated to ensure that the components displayed are as per the requirements, which also checks about the typo errors, and apart from that, the formats of dates, whether the numerical field is clearly mentioned, etc. Other than that, the field widths, which are limited to certain number of characters, also checked as mentioned in the data specification sheet prepared by the data management team. It also checks the layout of the CRF. For example, whether that needs to be a drop-down list or it should be a checkbox or radio button, so which is mentioned in the CDSS, it should be compatible to the same. Edit check specification. This also we discussed or learned in the earlier module, edit check programming, as well as in the ECRF designing module. So it is a document which will contain the programmed consistency checks which are used to minimize the logical possible error as per the study protocol. It can be either manually generated or system generated. System generated are nothing but which are automatically arised during the data entry process. These checks are programmed and uploaded into the system, and the error message will be generated that should be easily understandable for the person who is going to do the data entry. So it is always should be simple and easy to understand too. Validation procedure. The validation or testing is done by preparing a UAT log, because always a test scripts which are provided needs to be tested and same should be recorded in the UAT log. So it takes place in the test environment by creating some test patient and enter a dummy data. It should be both clean data as well as the dirty data to check whether the data passes or fails against the validity check. Why this is required? Because even for the clean data, there should not be any discrepancy. Once it is released to the system, for example, if a discrepancy arises for a clean data, then the programming is not done accurately. That is why we do the testing for both clean data as well as the dirty data. And also this validity check, which may be due to inconsistent data, missing data, range checks, and the deviations from protocol as well. So if any programming error occurs during the testing process, the same needs to be captured in the database testing error log. Global ruling. Global ruling is a document which states that queries can be closed by making changes in the data except critical fields without the consent of any other responsible stakeholders. Only the primary data which directly impacts the study cannot be altered through this document. For example, if a laboratory data is entered incorrectly, but if you have a source for the same, then the team can correct it and can provide the supportive document for future references. It doesn't mean that without getting any consent from responsible stakeholders, but it should have the supportive document for modifying the data, and that should be the source document. Flow of discrepancy notes. This section must contain information about how the query gets generated throughout the system, and it's assigned to a particular user, how the same is updated, and how it is finally closed throughout the validation process. Review of critical and non-critical data. Critical fields are reviewed manually so that to check the logical errors which may not be generated through the system. All data points are checked, and for critical data, that is 100% review is required, and that too manually as well. Non-critical data field is also reviewed, but as non-critical data is huge, the percentage of fields to be reviewed are as discussed with the sponsor. But critical data would be a minimal data, or those are actually critical data. So that has to be reviewed 100%. So data cleaning. Once the study is going to be in the final stage, it's to be cleaned, or if it's going to be a longer duration trial, that should be cleaned as per the frequency set process. So for data to be cleaned regularly, there should be a frequency set monthly or twice, or a manual review, which would be based on the listings pulled from the database, and these are mentioned in a document as agreed upon with the sponsor, followed by discrepancy management on a regular basis. Along with the manual review, the data cleaning should be completed. Unresolved discrepancies. Sometimes in a particular project, not all the queries would be closed or resolved. There may be some unresolved queries are listed. Those queries are to be identified and captured in this particular section. For example, there may be a patient query has been raised but discontinued from the study, and that has remained unresolved. For example, if the patient is discontinued due to any particular adverse event, but if you have a missing data field but the patient could not be reverted, so the data is empty. So there could be some discrepancy to that particular missing field. Those would be identified as unresolved discrepancy, because actually there is no any source data for the same to capture to the system. Finalization. The document is first created as a 0.1 version, or as upon their company's policy. It could be 1.0 or zero point 0.1, and that would be circulated internally for review. If any changes are suggested, after that the document will be updated and will be shared with an updated version of the same. So then it would be sent to sponsor and other stakeholders with the draft version of 1.0, or as per their project specific versioning or their company's SOPs. If any further changes are recommended, again the version number would be incremental. So once the sponsor approves, then the document would be finalized with the final version. Revision. If there are any revision occurs due to any changes in the protocol or study plan, or if anything is missed during the setup process, then a post implementation of the changes needs to be identified. Post production change request form comes in this section, and the same would be sent for sponsor approval as well. And in meantime, the DMP document also to be updated as per the revision, and the relevant guideline also to be updated along with the version history and date for the same. UAT, which is user acceptance testing, is an activity or process of validation performed by entering test data. That is, as I already mentioned, clean data as well as the test data should be performed for test patients in the test environment. This image is a format of UAT log which contains the test case ID. Every test cases is identified with a reference ID. This is nothing but a number or a character to identify each test subject with its unique test data, visit name or event name, which it will come under a specific visit. CRF name is nothing but in which form it is tested. Version number is the CRF version number which is used. Item name, which is nothing but the question prompt. And prerequisite, sometimes if a data should be visible or if a question should be visible based upon the earlier question the data, so that is called as your prerequisites. Prerequisite item data. So what type of data should be captured for the prerequisite item? And clean and dirty data, those are clean data or dirty data, or it should not be a missing data. Okay. Then data type. What is the data type? It is the format in which the value was stored in the database. Input is the test data which was used for validation purpose. So it would be clean data or dirty data too. And executed, when the UAT was performed and what was the expected result and actual result. Always the expected result and actual result should match, then only the testing process is valid. If not, then the same to be reprogrammed or revalidated again as per the issue. Then pass, fail, not executed or suspended is the status of the checks which are being validated in the test environment. Along with that, the person who tested the process, their initial also is important, so that in case if any issue arises, the same can be verified with the tester person. QC for user acceptance testing. QC activities are performed post finalization and internal approval, the sponsor approval if required. QC for UAT is done by randomly selected edit checks. Percentage may vary from study to study or sponsor to sponsor too. The error rate will be estimated using a formula, that is percentage error rate is equal to number of errors detected divided by total number of edit checks detected into 100. If errors are identified, it is reported in QC issue log and QC report for study, and also highlighted to the lead data manager or the project lead to take proper actions to correct the same. If any amendments are made to the document, it is revised and QC activities are performed again. Handling non-CRF data and data transfer guideline. So what is non-CRF data? Non-CRF data is a data specifically which is not entered directly into the system or collected in the ECRF. Those would be imported from the third-party vendor during the study, which includes central laboratory data, ECG, or any medical imaging data, etc. So how the same would be handled? The non-CRF data guideline is a document which deals with the activities that brief about the procedure required for handling of non-CRF data and to ensure accurate and timely import of data of each study. So it always states that at what frequency the document would be imported into the system. What is data transfer guideline? The data transfer guideline is a document which deals with the activities that brief about the procedure required for handling data transfer and to ensure accurate and timely import of data of each study. The requirements of non-CRF data handling and data transfers are data parameters, non-CRF data identification, format of data, medium of transfer, frequency of transfer, data transfer specification document, the calculated variables too, and key contact person who was involved in the transfer activity, validation, finalization, and revision of the. So the requirements. So data parameters. The data parameters required for the non-CRF data will be captured. Non-CRF data identification. So it will identify the non-CRF data as per the study, and also it will be defined in the CDSS and other guidelines as well. Format of data. So what type of format it would be? The file format. That also to be mentioned. And what would be the medium of transfer, whether it would be transferred through email or through CD or any other specific format, and what is the frequency, and whether the data is cumulative or incremental, that also needs to be specified. Then data transfer specification document also states the information regarding the file being imported, which will have the variable name, code formats, content of each file, etc. Calculated variables which allow you to create new data from the data which is already exist in your data source. Key contacts. The contact details has to be provided in the document if any assistance is required. Validation. The import of data to be validated as per the data validation guideline. Finalization and revision. So this document first should be internally approved, then only would be shared to the sponsor and other relevant stakeholder. Once it is approved by the sponsor, then final revision or final version is sent to sponsor for their records or for their signatures. So revision is required only if there is any change into the protocol or into your study plan or into your import sequencing, then revision should be made and circulated internally, then sent for sponsor. So once the sponsor approves it, then the DMP document will also be updated and the concerned guideline also needs to be revised with the newer version as well as the date which would be effective. Validation of data input. The clinical data analyst or the lead data manager shall validate the data input by executing the details such as medium of transfer, that is, whether it would be transferred via email or is there is any other specification of transfer, and the format of transfer, to whether it would be in the Excel format, SAS format, etc. in the test environment. If there are any errors found during the input of files, they are identified, and if any changes are required to be made into the guideline, the same will be updated accordingly. After the changes, the data should be imported again as per the procedure until it is done successfully. The data transfer specification document is prepared for each data input file and has to be signed off by both the parties who is going to send the file and who is going to receive the same. If the edit checks need to be performed on non-CRF data, it should be as per the data validation guideline. In case if the checks are not fired for a particular import, the data needs to be reviewed manually by the team. Non-CRF data review. When non-CRF data is imported from the vendor, not only the import is reviewed, but also the data need to be checked against the imported file. Non-CRF data is reconciled with key variables such as sponsor name, protocol ID, site ID, subject number, sample number, etc. In case of it is a laboratory data, or if it is an imaging data also, you would have some specific unique number for the same. The CDA, nothing but the clinical data analyst, also checks for any missing data is available. So if there are any discrepancies while reviewing, a query is raised in the database. The site has to respond to the same. Post response from the site, appropriate action is taken, like notifying the source if the discrepancy is related to the data received from the source. Then same would be communicated back to the source for clarification. The update can be further checked during the next round of reconciliations. The changes made in the data is again reconciled during the next import process. So for predefined ranges obtained from laboratories, programming can be done on these changes. If not done, then they has to be checked manually while reviewing. Validation of data transfer. So when the data is transferred, the same also needs to be validated. So the data transfer is validated by executing the details such as medium of transfers, and if there are any errors found during the transfer of the files, they are identified and changes are made in the guideline. So the data transfer specification document is prepared for each data transfer file and same needs to be signed off. Electronic data transfer form has to be updated with all required details before initiating any data transfer procedures. As already mentioned, the same for finalization and revision. So initially it would be in draft point 1, the version or as per the company's SOPs, the version control, how it is mentioned, same would be captured. And once it is shared to the sponsors, then the final version is released post their approval. If there are any comments, again the same would be updated in the document. Then it will be sent for the review and approval process. This cycle continues until unless it is approved. If there are any revision, then the DMP as well as the data transfer specification document needs to be revised or updated along with the version history and the date. Quality control. Quality control occurs at every steps, which we already discussed in the DMP, also in the quality control plan section. So QC activities are performed post finalization and internal approval, and a percentage error rate is calculated randomly selecting 10% or as upon agreed by the sponsor, and it also varies from study to study. Percentage error rate is equal to number of errors detected divided by number of fields, total number of fields checked into 100. If errors are identified, again same needs to be captured in the QC issue log as well as in the QC report. And LDM has to take an appropriate action until unless it is correct. So QC for non-CRF data handling, it's also again the same. QC activity will be performed post finalization and internal approval, and percentage error rate is also the same. And if there are any errors identified, again reported in the QC log and QC report as well. So as I already said, the same needs to be highlighted to the LTM to take proper actions to correct it. QC for discrepancy management. So again, it would be randomly selected percentage only, but this also varies from project to project. And again, percentage error rate formula is also same. If there are any issues, again reported in the QC log and QC report, and LDM needs to be informed about the same because they are only going to take the appropriate action and they're going to update the programming errors or the validation errors as applicable. QC for critical data. So as I already mentioned, QC for critical data should be 100% and that has to be reviewed manually also. If there are any error, and same to be identified in the QC log as well as in the QC report. Interim analysis. There could be interim analysis in some of the studies. Under such condition, QC will be performed for interim analysis too. So that is performed by randomly selecting the percentage of subject as upon agreed with the sponsor. And again, the formula is also same. If errors are identified, the same would be captured in the QC issue log and QC report, and LTM would be notified on the same to take further action on it. Final QC. This would be done at the end of the study before DB log. So it will be 100% QC of all CRF at least for 10 patients or as upon agreed with the sponsor. And again, error rate is the same. If any issues are identified, the same would be captured in the QC issue log and quality report. Thank you.